And we got a little bit into this packet. We began looking at balance here. And so far, we pretty much just described what balance means as it applies to the kidneys, what homeostasis means as it applies to the kidneys, and how our water inputs, which is diet, which is cellular respiration, must match our water outputs in order to keep our plasma volume concentrations constant, okay? And so based upon that conversation, we ended up here talking about this terminology. So normovolemia being normal blood volume. And although it's not written here, we want to also be thinking about solute. So if water is at an appropriate level in our plasma, solute is also at an appropriate level. So it's two sides to the same coin. Hypervolemia, on the other hand, being high blood volume, right? So a positive water balance state, so more fluid, in other words, less solute. And then hypovolemia being low blood volume, so a negative water balance, less fluid, and then more solute, okay? And then just going on here, we're going to speak about how water moves in more detail. A part of this will be review or repetition, but then we're really going to get into the loop of Henle, where a lot of water reabsorption takes place also. So just to kind of summarize here, we said most of the water reabsorption happens in a regulated fashion, in an unregulated fashion, that is across the proximal tubule, also across the loop of Henle. And so we talked about the proximal tubule mechanism. We're going to review it here briefly, but then what we're really more focusing on is looking at the loop of Henle mechanism for water reabsorption. And then a small part of that water reabsorption happens in an unregulated fashion. This is more so in the distal parts of the tubule, so the collecting duct and then the distal convoluted tubule. This calls on us to remember what osmosis is. Okay, so this calls on us to revise our definition of osmosis. So osmosis, we said, is water diffusing down its concentration gradient. In other words, that is passive movement. Water has to move from an area of low solute concentration, high water concentration, to an area of high solute concentration, low water concentration, okay? And so water reabsorption is usually following solute reabsorption. We made this connection we spoke on Monday as well. So in order for us to move water, and this is not just in the kidneys, this is across most of our epithelial surfaces, we have to create a solute gradient. We have to pump solutes first, and then water can follow, okay? On that basis, we then want to speak about the osmolarity of our body fluids and the osmolarity of our tubular fluid, which is our filtrate, starting out in the proximal tubule. So most of our body fluids we know is at an osmolarity of 300 milliosmoles. Another way of expressing that is that there are 300 milliosmoles of solute particles dissolved in 1 liter of our plasma, right? And so usually there's no osmotic force to move water. We know that water moves passively, but typically because all of our plasma is the same osmolarity, our tubular fluid starts out being at the same osmolarity. And so naturally in the body, there is no gradient for water to move passively. And so we're going to spend some time establishing a gradient, right? We have to set up these gradients sort of artificially in order for that water to then follow. Okay, so the kidneys are then going to compensate for changes in our osmolarity by changing how much water is reabsorbed. So what that means is that if my plasma becomes, excessively higher in osmolarity, so more solutes than water, the kidneys are going to reabsorb more water to balance out that plasma osmolarity. If my plasma ends up being too watery, so having more water and less solutes, the kidneys are going to reabsorb less water, again, in order to maintain that homeostasis, to keep my plasma osmolarity at 300. Okay, and so water is moving passively based upon an osmotic gradient, but kind of contradictory to that, we don't really have a gradient to start with. So the first thing we're going to have to do is establish a gradient, right? And that's kind of the next thing we're going to describe here. Just kind of highlighting those points here on this illustration. So we start out with our tubular fluid at an osmolarity of 300 milliosmol. It's the same osmolarity of our plasma. And so we made this point before when we talked about absorption across the proximal tubule. Because we start out with the blood being 300 milliosmol, the tubular fluid being 300 milliosmol, we don't initially have a gradient that's needed for water to move. And so what we do is we set up a gradient in order for that water to follow. Okay, and so what ends up happening is we end up having this medullary osmotic gradient. So the further down into the medulla we go, so going from cortex out here to medulla down here, we end up creating this gradient. Now we have a way higher osmolarity down at the very bottom in the medulla. And so we set up or establish this medullary osmotic gradient simply to allow this passive movement of water. So we just create the gradient that's needed for water to follow. Now on that basis, and we'll probably talk more about this later on, but we're going to see water moving passively across the descending limb and then solutes moving actively across the ascending limb. And that is how we actually create this gradient. This is really repetition, but I'm just going to go over this quickly just for the sake of repeating it. But we talked about the mechanism by which water is reabsorbed across the proximal tubule. It's not regulated. 70% of the water is reabsorbed here. And then across the distal tubule and collecting duct, most of the remaining water is reabsorbed as well. This area is regulated. So hormones are going to play a role here, mainly antidiuretic hormone or vasopressin. Okay. And then here's another sort of look at how that happens. So water reabsorption, we said, follows solute reabsorption. And so we mainly have to move solute first and then water can follow. So the primary solute that we're talking about is usually sodium, but there can be other things too. So potassium, chloride, or some of the other solutes that we can actively move and then facilitate that water reabsorption. So across the proximal tubule, we describe this idea that sodium is actively transported across the basolateral surface via the sodium-potassium pump. That established a gradient where we had sodium coming into the apical surface, other things hitching a ride with that passive movement of sodium, and then all of that creating a solute gradient, which then facilitates water movement. Okay, so this image here. And again, we talked about this before, but sodium moves in passively because it's being pumped out actively here. Glucose can hitch a ride with that energy. So this is secondary active transport. And then actually, let's go all the way down here and kind of summarize here. We talked about this before, so I'm not going to go into too much of the details, but we have some solutes moving in passively because they're actively being moved across the basolateral surface. We have other solutes hitching a ride, solute Y representing that hitching a ride, if you will. That is things like chloride, that is things like glucose, and then they have to be moved across the basolateral surface by facilitated diffusion. Okay, so it's also passive, but it requires a carrier. And then because of all of those things moving, we then have water move passively. All right. So that was the proximal tubule, right? Again, I'm not going to go into too much of the details of that because we talked at length about that in the previous packet. Here, let's get into what's happening in the loop of Henle. So that loop that's going down into the medulla, how is water being reabsorbed differently than what we see in the proximal tubule here in the loop of Henle? So essentially, we start out with the osmolarity of the interstitial fluid varying, right? So we have this osmolarity going from really sort of isoosmotic with our peritubular fluid up here, but then as we go down into the medulla, we have a much higher osmolarity, okay? And this gradient is crucial to reabsorbing water. So there's a lower osmolarity up in the cortex, and this is now looking at the interstitial fluid, so not necessarily what's happening in the tubule, but the tissue that's surrounding the tubule, and that is where we see the gradient that's needed for that water to move out of the tubule and into the surrounding vasculature. So we have to have this gradient where we go from low osmolarity out here in the cortex to a really high osmolarity down here in the medulla. And again, when we say osmolarity, we're just referencing the solute concentration. So we've got less solutes up here in the cortex, much more solutes down here in the medulla. And by having that gradient, which I'll talk about how we set up in a minute, by having that gradient, we now have the ability to move water where it's at a higher osmolarity, because our tubular fluid is at an osmolarity of 300, and now it's going to want to move out of the tubule into this much more concentrated interstitial fluid. And so we've set up the gradient for water to move passively. All right, so just reiterating those ideas here on this slide. And then I really want to stress what's happening on each region, each of these regions of the distal tubule. So down the descending limb, we only have movement of water. The descending limb is impermeable to all solutes. So there's no sodium movement, there's no potassium or chloride movement. It is only permeable to water movement. The ascending limb is the exact opposite. So the ascending limb has no mechanism for water movement. It's impermeable to water, but it does have solute transporters. So it's got a co-transporter that facilitates the active movement, so it's going to require energy, of potassium, of sodium, of chloride. So all three of those solutes are being actively pumped from the tubular fluid out into the interstitial fluid. And so that's going to be a part of how we set up this gradient. Also, we've got this urea transporter on our collecting duct. This is going to come into play a little bit later when we speak about the vasa recta, okay? But we want to make a note of this. So on the collecting duct, we have this urea transporter, specifically a urea transporter A, so UTA. And again, that'll make more sense later on, but this is facilitating the movement of urea from our tubular fluid into the interstitial fluid, and that is also going to be crucial in setting up our gradient, okay? And we'll talk more about urea later on, but urea is in our filtrates, in our blood. It's actually a byproduct of protein breakdown. So when proteins get processed in the liver, urea is what they are turned into along with nitrogen. But urea must be in our plasma so that it can end up in our urine to play this role, so that it can help to make the interstitial fluid here more concentrated. Okay. So let's talk about how we establish that gradient. But I'm going to pause here just to see what questions we have so far. Okay. All right. And this is usually kind of a tricky subject or topic for students. So spend some time with this. We're going to go over this several more times before we move on. But really spend some time with this. It may not make as much sense at the first, at first glance, but try to go over this several times. Try to draw it out. Try to make different ways of making it make sense. So maybe a table that helps you identify what's happening with descending limb, ascending limb, etc., or try to draw it out schematically. But just try to spend some time with this if it's not coming or not clear right away. Okay. All right, so we're looking at how we establish this medullary osmotic gradient. How do we end up having the interstitial fluid of the medulla so concentrated, having such high of an osmolarity? So essentially, we start out by using what's called the counter current multiplier. And the counter current multiplier is a really fancy word, right? I hate when we see this large, fancy, sort of convoluted terminology. And all it really means is that we have water moving across the descending limb and solute moving across the ascending limb. That's all that counter current multiplier means. So the descending limb only being permeable to water and not permeable to solutes, the ascending limb only being permeable to solutes and not being permeable to water. And having that counter current, right, that bidirectional movement is what helps us set up that medullary osmotic gradient, okay? So the result of the counter current multiplier is that we start out with a fluid osmolarity, so our filtrate, when it comes from the proximal tubule and it goes into the first part of our descending limb of the loop of Henle, it starts out with an osmolarity of 300. By the time we get down to the the bottom of the loop of Henle because we have lost water, right? Because we only are permeable to water across the descending limb, all of this water leaves our filtrate. And so now the osmolarity of our filtrate down at the dip, down at the deepest part of the loop of Henle is much higher, okay? So the reason is because we're losing all of that water. The descending limb is only permeable to water. And so the osmolarity is going to approach, it's going to sort of equalize with the interstitial fluid osmolarity, and it gets higher as we go down the descending limb, right? The osmolarity increases as we go down the descending limb. Looking at the ascending limb, we see that the opposite is true. The ascending limb is only permeable to solutes, so not permeable to water at all. And so the osmolarity here is going to get increasingly lower. So as we move all of those solutes from our filtrate, so now we had a really high concentration of solutes here, and now we remove all of those solutes, we now see that the osmolarity goes in the opposite direction. It gets less than it is in the interstitial fluid, but it also gets less than what we saw in the descending limb, right? It gets lower than it was in the descending limb. And so that is the result of our counter current multiplier. Having fluid only move on one side, solutes only moving on the other side, is ending up giving us this high solute concentration of our filtrate down here at the dip in our loop of Henle, but then having a much lower solute concentration or a much lower osmolarity when we get back to the end of the ascending limb of the loop. Okay.